Hi everyone, and thank you very much for joining today's webinar of our Discover Molecular Interactions webinar series. This is actually already our third season of this webinar series, so if you're joining for the first time, make sure to check out our YouTube playlist to find other interesting topics in our previous episodes. And today we will explore the importance of target density when developing targeted therapeutics, like, for example, antibodies. And I'm very happy to introduce today's guest speaker, Nichole Cerutti. Welcome, Nichole.
Thank you. Hi, Vivian.
Nichole is the Director of Biophysics at RoukenBio, and she's currently focusing on how biophysical methods can aid in the development of therapeutic molecules. If you have any questions during the talk, please feel free to use the Q&A function, or if you don't see that, you can of course also use the normal chat function. Or alternatively, you can also raise your virtual hand afterwards, after the talk in the Q&A session, and I can unmute you and you can pose your question to Nichole directly. And now, without any further ado, I will hand over to you, Nichole, and I'm looking forward to your talk.
Thanks, Vivian. I'm really happy to be here and looking forward to sharing our study with you. I'd like to first, though, take the opportunity to introduce RoukenBio. We are a CRO, a contract research organization, and we provide drug development services and support to a global customer base. And these customers are developing small molecules, large molecules, as well as advanced therapeutics, from early discovery all the way through to clinical phases. We provide discovery to all the way through, rather, to IND enabling immunology and bioassay research services in the areas of inflammation, in autoimmunity, in immuno-oncology, and also oncology. Our services span immune cell assays, cell line engineering, safety and efficacy assessments, as well as mechanism of action studies. Our company was founded in 2015, and we were known then as Antibody Analytics. And we began by specializing in monoclonal antibody characterization, in particular, for those customers who were specifically developing biosimilars. The initial focus was measuring antibody effector function in complex matrices. And as we grew, we integrated biophysical services by acquiring 2 BioCore 8K systems. We also added advanced immunology services with primary cell models, human cell models. And we also then launched our innovation department in 2019, which allowed us to drive new bioanalytical capabilities. By 2022, we introduced cell line engineering and filed our first patents, which culminated in the launch of our Index 2 platform in 2023. And following this, we had our first external investment and allowed us to recently rebrand to RoukenBio. And this reflects our broader focus as a discovery-centered CRO. Our newly renovated Discovery Center is located just outside Glasgow, and we have just over 100 staff members. And this is what allows us to offer a unique and collaborative approach to drug discovery. And this is grounded in scientific empathy. We're on a mission also to be one of the most trusted research partners for biotherapeutic innovators. And we are aiming to deliver quality data, which will accelerate drug discovery. So while traditional kinetic assays like SPR, which is what we've always been offering, have typically used isolated proteins or simplified systems, these type of assays don't always fully capture the complexity of a real cellular environment, where there are factors like receptor density, membrane and receptor dynamics, and cellular context, which can all collectively or individually significantly impact the binding behavior. Although our key biophysics offering has centered around SPR, we've had an increased need for the ability to measure kinetics directly on cells. By measuring kinetics on live cells, we are able to gain a more accurate picture of how a therapeutic interacts with its target under native conditions. And this allows us to account for binding avidity, things like receptor clustering, and also cell-specific factors. And these factors can influence both the strength as well as the duration of binding. Ultimately, cell-based kinetic measurements provide insights that are much closer to what we'd expect to see in actual biological systems. And it makes this data highly relevant for predicting therapeutic efficacy, for optimizing dosing, and also for minimizing off-target effects, which is what you see in real world conditions. So this brings me on to single cell interaction cytometry, which I'll refer to as scIC. And this technique we were made aware of when we first came into contact with what was then known as dynamic biosensors, and that was about 2 years ago. In scIC, target expressing cells are injected into a microfluidic channel, and cells are trapped in nanocages attached to the biosensor. So you can see here on the biosensor surface, there are cages there that trap the cells. Following this, your fluorescently labeled molecules are passed over the cells, and the association of the therapeutic with its target is measured in real time. Once equilibrium or steady state binding is achieved, the fluorescently labeled molecule is replaced with buffer, and the dissociation of the molecule from the surface can then be monitored. So as with SPR, there's a sensogram trace where the typical response units that you would measure in SPR are replaced here with fluorescence. But you can see here there's an increase in concentration of an analyte passed over the cells that have been captured, followed by dissociation of the analyte from the cells. The chip is then regenerated by pushing the cells out of the traps with a reverse buffer flow. So knowing that we can offer SPR, flow cytometry, and now recently scIC, I'd just like to highlight the differences between the techniques. So with surface plasmon resonance, you have your target immobilized, and this target is an isolated target, a recombinant protein in most instances, and you're able to monitor in real time the binding of your specific molecule to the target of interest. Challenges here, and this is why we looked to get the scIC instrument, is that cells can theoretically be immobilized on the surface, but oftentimes this immobilization is very unstable, and most importantly, there's limited detection depth. So although you're capturing a cell, you're only really looking at your molecule binding to a very small portion of the cell, purely because of the resonance able to not being able to be detected past a certain area on the chip surface. Flow cytometry, again, is a very good way to measure KDs. However, that's all it is you're able to measure KDs. You're not able to decipher the rates involved in that. So it's an endpoint analysis telling you how strong the binding is, but you're not sure why the binding is measured at that particular level. There can also be very tedious workflows involved with flow cytometry. So going with the single cell interaction cytometry is a combination of the principles behind the SPR as well as flow cytometry and combined in one. And it seemed like a really, really great technique for us to implement and use for our customer studies. That brings me on to one of the key challenges for developing tumor targeting therapeutics, and it lies in identifying optimal tumor-associated antigens. Often you find that these target antigens are not cancer-specific. In other words, they're overexpressed on both cancerous tissue and in healthy tissue. So the heterogeneous tumor antigen expression, not only makes efficient treatment more difficult, but it can also contribute to immune evasion by tumors. And these tumors are down-regulating the expression of target antigen, which is what we call antigen escape. This challenge applies to multiple immuno-oncology therapeutic modalities, including molecules that are immune cell therapies, immune cell engagers, ADCs, and conventional antibodies. The expression of target antigens in normal tissues can also lead to off-tumor toxicity, and this is a common side effect associated with many immunoagent therapeutics. So in particular, on-target off-tumor toxicity has become a significant concern, and regulatory guidelines have been established to assess the undesired targeting of healthy tissues that express the target antigen. The conventional approach to assess the impact of antigen density on the efficacy of therapeutics is the use of a panel of cancer cell lines. And these cell lines will be expressing different levels of the antigen of interest. However, there are multiple limitations with this approach, and these limitations include cost, access to multiple cancerous cell lines, the expression range, as well as the cell background. So RoukenBio has developed a solution to this, and this is our Index 2 system. It's a platform which utilizes chemical proximity systems for the inducible and titratable expression of one or two target antigens, and this is done in a very controlled manner. We've built our Index 2 system using CHOK1 as the background and introduced an induction cassette, and then a second construct which contains the target or targets of interest via lentivirus. So to exemplify the system, we've used a number of target antigen examples, and we've been able to demonstrate, as you can see in the graphs in the middle of the slide, that we're able to control the expression of antigens over a wide expression range by varying the concentration of inducer. So these are four of the examples for the single antigen. We've got HER2, EpCAM, BCMA, and CD19. And you can see with increasing concentration of inducer, we get increased expression of the target antigen on the cell surface. We've also been able to show this with the dual expression, and we're able to simultaneously induce the expression of two different antigens. And these are also at varying levels, and again using exactly the same system by controlling the concentration of the inducer. The only difference here is that each of the receptors that are expressed are controlled by a different inducer. The CD22 in this example is under the control of indole-acetic acid system, and the CD19 under the control of the abscisic acid system. So we then sought to see how we could integrate the use of this novel inducible cell line expression system with the scIC using the Helix Cyto instrument. And the main question or point of interest that we had was, what are the effects of target density on the kinetics of antibody binding? So what we did was choose 4 differentially expressing HER2 cell lines, and we quantified the expression of the receptor by flow cytometry, just using Bangs beads and Bangs beads and calibration. And we went with low, medium, and high expressing cell lines, and we fixed these cells because these cells were sent through to Brooker, and they have run the experiments for us in anticipation for receiving our instrument. So that's how the study was performed. So trastuzumab was fluorescently labeled, and the fixed cells expressing the specific level of HER2 were passed over the surface of the chips. And this was followed by 3 increasing concentration of trastuzumab So trastuzumab was used at 1, 5, and 25 nanomolar. And then this was followed by an extended injection of buffer, which allowed to follow the dissociation. Trastuzumab was able to bind all 4 of the target expressing cell lines. And as a control, we used the wild type CHOK1s that had not been induced or transduced. And then we had the CHOK1s that had been transduced but not induced. So you can see they were very good controls for this. What you can quite clearly begin to appreciate when you just take a look at the sensorgrams is that there seems to be a difference in the way that the molecule, the trastuzumab, is dissociating. You can see just by looking at the cells which have the very high number of receptors versus those that have the lower number of receptors, that there's definitely a difference in the way that the molecule is coming off the surface. The nice part about the instrument that we found is that you're able to, the instrument comes with a light microscope, so you can see how many cells have been captured when you're looking at the binding interaction. This allows for the data to be normalized to a per cell binding level. So you can normalize the fluorescence signal to the number of cells. And for our study, you can clearly see that the more receptors you have on the cell surface, the higher the amplitude is. In other words, the more receptors means you have more binding sites for trastuzumab, which means that you have a higher signal that you're able to measure. And this is where the really cool stuff comes in. When you fit the data to a 1-to-1 Langmuir model, you can see here that the KDs measured are all in the picomolar range, where the cells with the lowest receptor number have the highest KD and therefore the lowest affinity, whereas cells that have the highest receptor numbers have the lowest KD and therefore the highest affinity. And here's where we can start to really tease apart what's causing these differences. When you look at the on rates over here, you can see that they're all very similar, irrespective of the number of receptors per cell. However, when we look at the dissociation rates, the number of receptors per cell starts to play a very important role, with presumably avidity effects. Where with the very high numbers of receptors per cell, you can see the half-life has increased almost tenfold to 30 hours, as opposed to those cells which have the very low receptor numbers, where you only have a half-life of 3 hours. In other words, the take-home message from this is that the off rate is driving the differences in the KDs. So that's the biggest study that we've done so far with our instrument. But I'm sure your imaginations have started running wild, thinking about all the different iterations and things that we can try. And so in our innovation team, what we are looking at studying is internalization and recycling, and also knowing that we have the ability to finely titrate the expression of receptors, especially 2 receptors. We'll be looking at the binding of bispecifics to this inducible cell line system using different expression levels of the different receptors that are targeted by the bispecific. Yes, hopefully these applications will allow us to open new possibilities for designing more sophisticated and durable therapeutics with targeted engagement strategies. Thank you very much for your time, and I hope you enjoyed my talk.
Thank you very much, Nichole, for these great insights. Yeah, we are now taking questions, of course. So as I said before, you can, of course, talk to us directly by just raising your virtual hand and I will unmute you. Or you just put anything you want to ask in the chat or in the Q&A and I will forward it to Nichole. So I will start with the first question. Since we have talked a lot about density now, is there any any detection limit with regard to the target density on the surface?
With regards to detection limit, um, as you can see with our study in particular, we went down to 6,000. However, I think there are no limitations. It would be dependent on the number of receptors, their affinity. Obviously, the higher the affinity, the more molecules you'd see binding, even if you had low target numbers. And also with the chips, what you have available is chips that capture five. There's 5 different cages on the actual electrode, so you're able to then amplify the signal by 5, if that makes sense, as opposed to those chips that are available with the 1 cage. So I think there's multiple things to try when it comes to target expression.
I see. Thanks. There's a question on your methodology to fix cells. So you were mentioning that you fixed the cells before the measurements. Could you comment on the method you used for that?
Um, yes, I can comment on the method, but it's slipped from my mind. It's the method that we use for fixing the cells was a standard method that was supplied to us by dynamic
Mhm.
biosensors, Booker biosensors. Can you remind me, Vivian? Can you remember the formulation? But I
So
think it's on your website. If
I'm
I'm
pretty sure we usually use PFA.
Yes,
I
that's it.
cannot comment on the exact concentration, but I can look it up and send you a message afterwards. I mean, the
Yeah,
standard I think is like 4% PFA,
yes, that rings a bell.
but
That's it.
for like the correct like incubation time, temperature, I'm happy to get back to you on that to
Yeah,
answer your question in
yeah, I
more
seem to
detail.
I seem to remember it was done at room temperature. I just couldn't remember PFA. But it's just a standard self-fixing. We
Mm,
are aware as well that you can use, there are certain reagents that can also block internalization, because that's one of the
mhm,
reasons why people do fix cells. And that's just a chemical treatment that also stops molecules from internalizing. So there's also the option to do that.
true. Yeah. Then there's a question about if scIC can be used for yeast cells expressing antibodies on the surface. Now, I'm not an expert on yeast cells. Could you maybe tell me the size of yeast cells? Because we do have size limits for scIC. Um, I don't know, Nicole, do you
I mean, so
know?
as I'm aware, any mammalian cells, be it adherent or a suspension. So I would assume, as you say, depending on the size of a yeast cell, that
Mhm.
would really be your limitation. There we
Yeah.
go, 5 to 10 micrometers. So yes,
Yeah, that would work because we have like a lower limit sort of. We have different sizes of traps depending on the width of the traps, which can accommodate for different cell sizes. So there's like L traps if you have very huge cells from like 20 to 25 micrometers, but then there's also M for like medium-sized cells, and then there's the S traps that would accommodate for roughly like 6 to 14 micrometers. So that should work fine. So if you have a project about yeast cells and you want to give it a try and you just want to find out whether your yeast cells work with it and survive the process of the kinetic measurement, please just shoot us a message and we are always happy to organize a demonstration at your lab and just test out your cells. Um, then there's a question, could you comment on how receptor internalization and recycling would work in this context?
yes. So in terms of internalization and recycling, typically what's used is well, the very simple way to do it is to simply um, have cells that express the neonatal receptor. For example, if you were looking at recycling, which would use the neonatal receptor, or there's using cells that just would via pinocytosis engulf or internalize the molecules. And the simple way to do that in normal cell culture would just be doing, collecting cell culture media so that you can quantify the amount of antibody that is being internalized and non-internalized, and staining for antibody that's still on the surface. However, with the advent of pH sensitive dyes, um, one way to do this is to label the molecule with a pH-sensitive dye that changes fluorescence from green to red or red to green, which is something that would happen upon internalization as the pH of the lysosome is decreased. You would presumably, this is the hypothesis for us anyways, you would presumably see the change in the fluorescence because the instrument is able to measure a dual fluorescence, so red and green. I don't know what the data would look like, if I'm honest. Something would be appearing and something would be disappearing. So as I say, this is something that's in our pipeline, but that's really the hypothesis around how we would design an experiment like that.
Thank you. Then there's a clarification for the question about the fixation. I might have understood that wrong. So I was talking about the general fixation of the cells, but the question might rather have been about a fixation to the surface of the chip. So we don't fix cells to the surface of the chip. They are just physically retained like in a soccer goal, so to say. There's no fixation to the surface. You don't need any anchorage molecules or capture molecules. It will just be flushed in and stopped physically by the cell trap. I hope that clarifies it. There's another question, how we measure the receptor density for the type of cell you are using.
So this is Bangs beads. That's what we use. There's lots of different ways, beads that are available. So either Bangs beads or the QuantiBite system, and that's basically beads are supplied with a known concentration of antibody on their surface, and a known you therefore are able to see at a certain level of fluorescence. How you are then able to get a measurement for a standard curve, and then you do the same thing for the cells that you're expressing the receptor levels on, and you're able to then look at the standard curve and figure out exactly how many receptors are being expressed per cell.
Thank you. Okay. Are there any other questions or follow-up questions? Also, if you come up with another question after our webinar, please always feel free to reach out, either to Nichole or to me, or to just the company via our website, our contact forms. We are always happy to clarify our technologies or also discuss your specific projects, your specific research questions. So don't hesitate to reach out. Because there's the question about the presentation. So there's definitely going to be a recording of the presentation be shared, and we will also send follow-up emails. So you will receive some follow-up information on this.
I think there's a question.
Yeah, about SPR. So mass transport, considering the amount of receptors, whether you should choose the mid-level rather than...
It's a very important question, actually. And simply put, what you need to consider is in SPR, you are very much dependent on the size of molecules because of the way that the light is resonated, whereas this is fluorescence. So it's irrespective of mass. It's purely fluorescent, so it's a very different kind of phenomenon. Although it's done on a biosensor, you're using fluorescence rather than a change of mass per se on the surface. I think that's the question that you're answering, if that helps.
Okay. Well, then I think it's a very interesting, also not only considering that you want a lot of avidity effects, it's also sometimes the other way around, right? I mean, sometimes you want a rather smaller dwell time for CAR T cells, for example. So like being able to tune the off rates and looking into the effects of target densities in that respect, I think it's a very important topic.
Yeah, we do use these cells in assays where we're able to determine the threshold for activation. And that then speaks to what you've just mentioned there, being able to understand how much of your molecule is needed in order to exert its effect so that you can take these things into consideration.
Mhm.
Yeah.
Yep. Great. So thank you so much for your presentation and the discussion, Nichole. And thank you all for joining us and for
Thanks
your
for having me.
questions. So, yeah, stay tuned for next episodes of this webinar series. I hope to see some of you in our upcoming episodes, and I wish you a very great rest of your day, and goodbye.